Hello, my name is Louis Ken Rangel,
My name is Cassie Olvera.
and today we are here to discuss our research. As the title suggests, we are discussing the synthesis of aminocyclobutanes and methylaminocyclobutane derivatives with potential use in CNS drugs. So, CNS drugs are drugs meant to target the central nervous system. In our research, we are specifically discussing NK3 antagonists and the glycine transporter 1 inhibitor, which are meant to target schizophrenia, depression, and Alzheimer's. So these drugs are meant to target this, but the main issue with current drugs on the market right now is that a lot of them cannot cross the blood-brain barrier. What is the blood-brain barrier? It is an unavoidable obstacle that any CNS medication has to interact with. What we are posing is by using the potassium 3 component reaction, the drugs will be a lot more permeable through the blood-brain barrier, allowing it to overcome two main obstacles. The drug itself will be more effective, and also there is a lower chance of neurotoxicity, because a lot of the time you think the medication doesn't work, we're going to up the dosage, and that leads to neurotoxicity, which leads to death. So hopefully by using this new mode of synthesis, the potassium 3 component reaction, we can prevent that from happening. So why the potassium 3 component reaction? Because the potassium 3 component reaction allows us to vary the type of carbonyl, amine, and boronic acid we're using. Specifically for our research, we have varied the boronic acid to the 4-fluorophenylboronic acid, which has allowed us a significant higher yield. Also, the potassium 3 component reaction has a higher stereoselectivity for functional amine formation, allowing for the drug itself or the final product to be more effective. And one of the main drawbacks of using the potassium 3 component reaction is the necessity of requiring molecular sieves, which remove water from the reaction, allowing it to run more effectively. To talk more about the mechanism and also our trials is going to be Cassie.
The mechanism is believed to start with the reactive carbonyl group and a secondary or a primary amine. And it's also believed to go through two different routes because it's not fully understood. And one possible route is the formation of the amine to aminium ion and then undergo a coupling mechanism with the boronic acid. And the second alternative method that is believed to undergo is either where the carbonylamine forms a boronate complex with the boronic acid. And both of these routes are believed to form a secondary amine or a tertiary amine. So the reactions we have first started involved a primary amine, and methods involved refluxing and molecular sieves, but based on GCMS and crude proton NMR, there's no evidence of the product being formed. And so further research was conducted, and using a secondary amine showed a higher yield through a potassium reaction. And we performed the reductive amination reaction to synthesize our own secondary amine, and this secondary amine was combined in methods of molecular sieves and also a combinational method with molecular sieves under microwave conditions. And proton NMR and a LCMS was used for the molecular sieves to confirm if the product had formed, and for the second reaction, where molecular sieves were used under microwave conditions, a proton NMR confirmed the formation of the tertiary amine. The reaction that was used with molecular sieves resembles a significant peak at HI, which is a singlet at around 5.1 ppm. And this confirms the formation of the tertiary amine. And the LCMS that was recorded with the same reaction falls with signaling at peaks that were predicted at 362 and 363. The pure proton NMR that was used with molecular sieves under microwave conditions again showed the significant peak at HI, which is a singlet at 5.1 ppm. Current progress involves removing the residual starting material, which is seen as a acetaldehyde, where it has been still visible even after further purification, and we have tried to remove it with sodium borohydrate and methanol. However, that has not been successful.
So our future studies hope utilizing different types of boronic acids, potentially finding an even better acid than our 4-fluoroboronic acid, and also synthesizing different types of secondary amines to see the potential of a higher yield and also just a better stereoselectivity of our product. That is the end of our presentation. Thank you so much and have a great rest of your year.